In this video lecture, I'm going to cover codominance, which is another form of inheritance of some traits. So for codominance, co means together. So we're no longer looking again at like a dominant and a recessive the same way that we were looking at them with monohybrid crosses. Now we're going to have two alleles that are dominant together. So when we inherit both of the different alleles, when we're heterozygous this time, we're going to have a third phenotype come about that is going to be an equal mix of what we would normally classify as the dominant and the recessive allele. So for instance, like if our normal dominant allele is having white fur and our recessive is having black fur, the heterozygous would have black and white fur, because technically our dominant and our recessive alleles are equally dominant. They're both going to show. So we really don't even have a recessive allele anymore. Some other silly examples, right? Beaver crossed with a duck would get you a platypus. Shows the traits of both beaver and a duck together. This is just a silly joke, right? Horizontal stripes crossed with vertical stripes would get you a heterozygous phenotype of both horizontal and vertical stripes. So incomplete dominance, the heterozygous phenotype was a blending. It's like black and white would have made gray. Codominance, where each of the different traits are dominant together, black and white are going to produce a heterozygous phenotype of black and white. So some examples of this in real life. In cattle, you can have cattle that have red fur, you can have cattle with white fur, and you can have a heterozygous phenotype that's called roan, which means that there's red and white fur together. In some flowers, we can have pink petals, you can have white petals, and the heterozygous phenotype is going to have pink and white petals. In chickens, we can have chickens with black feathers, we can have chickens with white feathers, and we can have chickens with the heterozygous phenotype having black and white feathers. Again, in cattle, we can have cattle that have black fur, cattle with white fur, and the heterozygous phenotype would be black and white fur. In sickle cell anemia, which is a genetic disorder, if you are homozygous, you will either have all normal red blood cells that are these donut shape, or you can have all your red blood cells be this sickle-shaped, which is fatal. If you're heterozygous, you have both donut-shaped red blood cells and sickle-shaped red blood cells. And what's an interesting aspect to this is actually having these the sickle-shaped red blood cells makes you resistant to malaria. So both of these two would be resistant to malaria, which is that disease that mosquitoes carry that's very deadly. But this heterozygous phenotype here, not only are they resistant to malaria, but because half of the red blood cells are normal, it's not a fatal disease, whereas all these sickle-shaped red blood cells, yes, they're resistant to malaria, but the sickle-shaped red blood cells clot easier, so it's fatal. So looking at a problem, we're going to do these problems the exact same way we did for incomplete dominance, where we're going to use two different letters to represent the alleles. And we're going to do step one the same way we did with incomplete, where we define all our genotypes. So in cattle, red hair and white hair are codominant, with the heterozygous having roan hair. A white cow is crossed with a roan bull. What are the genotypes and phenotypes and their percentages of the F1 generation? So I told you to use R for red hair, so red hair is going to be represented with a RR. White hair will have to have two W's, and heterozygous would be RW, and that would have roan. We're crossing a white cow, so that's going to be WW, and a roan bull, which is going to be RW. And we can plug that into our Punnett square and solve. The top row will give us all RWs, and the bottom row will give us all WWs. For our genotypes, we have 2 out of the 4 boxes being heterozygous, so that's 50%, and 2 of the 4 boxes having WW. Again, we can't just say homozygous because we aren't specifying whether we're homozygous for R or homozygous for W, and we can't say dominant or recessive because there aren't any dominant and recessive alleles anymore. They're both dominant. For the phenotypes, my 2 heterozygous genotypes will code for roan hair, and my 2 WW genotypes will code for white hair, so it's going to be a 50-50 split. For our second problem, we're working again with cattle. Black fur and white fur are codominant, with the heterozygous having both black and white fur. A black cow is crossed with a black and white bull. What are the genotypes and phenotypes and their percentages for the F1 generation? So let's define our genotypes. Black fur is represented with the B allele, so you need 2 Bs to have black fur. White fur is represented with a W, so we would need 2 W alleles to have white fur. And the heterozygous phenotype or genotype is going to be BW, and that will code for black and white fur. We're crossing a black cow, so that's going to be BB, with a black and white bull, which is going to be BW. Plug those into your Punnett square and solve, and this is what you would get. For genotypes, this top row is going to be, just leave it as BB, so 50% will be BB, and this bottom row is heterozygous, so the other 50% will be heterozygous. And then for the phenotypes, BB is going to code for black fur, so it'll be 50% black. And then BW will code for black and white fur, so it'll be 50% black and white fur. For the third problem, why don't you pause the video, see if you can solve it yourself, and hit play when you're done. So for this one, we have camellia flowers that can have red petals or white petals. Heterozygous would be red and white. We're crossing 2 red and white plants. So defining our variables, red petals would be RR. Our heterozygous would be RW, and that'll be red and white petals. And then WW will be white petals. We're crossing 2 red and white petaled plants, so that's going to be RW by RW. Plugging those into our Punnett square, these are the babies that you should have gotten. For the genotypes, it looks like we have a 1 in 4 chance of the baby plants being RR. That's 25%. 2 out of the 4, or 50%, will be heterozygous. And 1 out of the 4, or 25%, will be WW. For phenotypes, that RR is going to code for red petals, so it'll be 25% red. The heterozygous will code for red and white petals, so that's going to be 50% red and white. And then the last 25% will be WW, so that will be 25% white. And that's going to be it for codominance.